Hello and welcome to this special PIR live event webinar. Thanks for helping us kick off Science Literacy Week 2017. Thanks also to Science North, Helios Makerspace, Concordia University, and Partners in Research Canada for making this event happen today. My name is Stacey Joyce and I will be your host. If you're joining us live, remember that you can ask questions anytime by clicking chat in the top left-hand corner of your screen. Make sure that you adjust the to field and select all panelists and attendees, and then we will make sure to relay those questions to our guest expert later on. Please make sure you include your name, your school, or your city when you ask each question so that we can give you a shout out. Today's webinar is extra special. We have over 1,600 students joining us, including 7 groups joining us from video, all from locations across Canada, coast to coast. Let's meet our live classes, starting in the West. We have Mrs. Smith's grades 6 and 7s in Comox Valley, BC. Let's have a hello and a wave. I think they're ready. Next up, we have Mrs. Allen's grade 4s and 5s in Sechelt, BC. Hi. Welcome, thanks for joining us. We now go to Calgary, where we have Mrs. Wessel's class. And we also have, I'm just looking here, we have Ms. Burkell's class in Calgary too. And then we go all the way to Montreal, where we have, I think we might have some of our students. I know they were coming in from lunch a little late. And there we have a few people in there already. Hello to Madame Giroux's class. All right. Bonjour. Bonjour. Bienvenue. And then we go all the way to the East Coast. We have Madame Brunet and Mr. Thorne's students. Welcome. Awesome. Okay. And I believe I've hopefully grabbed everyone except our main location here. We'll finish off there at Science North in Sudbury, Ontario, where we have over 200 students joining us. Hello, Science North at the Valley Cavern. I don't think you're excited enough. You're going to have to ramp it up for this, I think. And okay, so we have now had a peek at everyone who is joining us across Canada. We also have a very special DNA expert today at the Valley Cavern in Science North. We have Dr. Thomas Merritt and his helper Lila with us today. And Dr. Merritt is a professor and Canada Research Chair in Genomics and Bioinformatics at Laurentian University. Thank you so much for joining us today. I will let you take it away.
Thank you, everybody, for joining us from all over Canada. I'm attached to my daughter. This will be a great event. I've been teaching genetics for about 20 years, 7 of them with my daughter. And one of the most interesting challenges when you try to engage people in genetics is you don't get to hold on to it. You don't get to touch it. You don't put it in your hands. And what we're going to do today is actually start right here. Everybody hold up your strawberries. And by the end of the day, when your hand is up, your hand's going to have DNA in it. You're going to be holding that molecule. And the really amazing thing to me as a geneticist is the same molecule that we're going to pull out of that strawberry in your hand is exactly the same molecule that makes you, you. One of the amazing things about genetics, about biology on this planet, is we all share the same informational molecule. It's DNA. And at the end of the day today, you're going to be holding that DNA molecule, and you're going to start with that strawberry, and you're going to finish up with DNA. And that DNA makes that strawberry, that strawberry, and that DNA makes you, you. The reason that I have blue eyes, and the reason that my helper has blue eyes, is our DNA. And the really amazing thing to me is the fact that Lila's beautiful blue eyes are a slightly different blue than mine, because she has DNA from me, and she has DNA from her mom. And we can actually begin to understand how that works. And the first step in understanding how that biology works, how genetics works, is understanding the DNA molecule that you've got in your hand. So we're going to start with a strawberry. Who knows where the DNA is in that strawberry? You, yes. Wait, is it inside the seeds? That's a good question. It's inside, but not just inside the seeds. Somebody else? Yeah. It's inside the juice? No. Think small. What? Okay, you in the back, jumping up and down. Script, yell it out. It's inside the cells. So you're going to start with that bearing in front of you, and we've got to get the DNA out of the cells. So what's the first thing that we have to do to get to that DNA? Yeah. We have to smush it. So just give me a second before you start smooshing. What we're going to do today is we're going to start with that berry. We're going to break that berry up, and then we're going to break those cells up, and we're going to release that molecule. That DNA molecule is so incredibly thin that even with the most powerful microscopes that we have on the planet, you cannot see that molecule. It is that thin. Does anybody know how long the DNA in a single human cell is? One cell. Yeah, yell it out. No, we'll wrap around the world. Every cell in your body has 2 meters, 7 feet of DNA. That strand is so incredibly thin that you cannot see it with a microscope, and it is 2 meters long. In that strawberry in front of you, there's the same kind of DNA. There are thousands and thousands and thousands of copies of an incredibly thin strand that's 2 meters long. And what we're going to do is burst those cells, and all those strands are going to spread out in the liquid. And then we're going to put another liquid over the top, and it's going to form a cloud. It's going to be called precipitate. We're going to extract the DNA. So we're going to start with the DNA in the cells, we're going to burst the cells, and then we're going to precipitate that DNA. And the fact that it's so incredibly long is why this is going to work. So now, let's all be geneticists. Hold your berries up and mash away, please.
All right, then, that's pretty loud mashing. I'm just gonna mute that for now, but I think we might be able to check in and see who else is maybe mashing up some fruit. Why don't we start on the East Coast now? How's it going there in Newfoundland with Madame Brunet and Mr. Thorne's students? How's the mashing?
Strawberries. Very big burst.
And is it strawberries that you're mashing?
That's the strawberries right now. Yep.
Excellent. All right, good luck. Let's check in now with Ms. Burke-Hell's class. How are things going there in Calgary?
Good.
Amazing. Beautifully.
Are we mashing strawberries or something else or a couple things?
Grapes.
Grapes? Grapes and
bananas.
and bananas. We'll have to see. We'll have to see later how they're different. Okay, I'll leave you to your mashing. Okay, let's check in with Mrs. Allen's class. I see some students there ready to show us. What are you mashing there?
Bananas and
It's pudding.
I put mine near the bottom.
Looks like pudding.
It does look like pudding.
It does. If we got the whole bag filled up, that would definitely look like a lot
Smoothies
of it.
on me, guys.
Yay!
All right, excellent. We have banana pudding-like substance in BC. Let's see, how are we doing in Montreal? How are things going there? At École Lovive, how are we doing there? C'est bon?
Oh, hello.
Hello.
Hey.
Bonjour. Are we mashing strawberries?
Yeah. Bonjour. Ah, bonjour tout le monde.
Bonjour. Are you mashing strawberries today, or did you choose a different fruit?
Strawberries.
Excellent.
Getting started.
Okay. Let's go to Mrs. Wessel's class in Calgary. Hello. Mrs. Wessels, are you using strawberries or bananas or something else?
Bananas. We're using bananas today. I tried strawberries though, and they work really good.
All right. Okay, maybe we should go to BC for Mrs. Smith, and then we're going to jump back to Science North and see how it's going there. Mrs. Smith's class, are you doing strawberries today, or did you choose a different fruit?
Blackberries and bananas.
Excellent. So we have something to compare today. We'll check in with you a little later. Okay, how are we doing at Science North, Dr. Merritt?
All right, so that, actually, Stacy, that was a really great point. So some of us are using strawberries, some of us are using bananas. We're using other things. Maybe we've got blueberries. Who's got the blueberries here? Hold up the blueberries. Blueberries in the back. So one of the really awesome things about this experiment is it works with any of the fruit that you want. And you can actually do it with spit. We're not doing it with spit today, but you can actually extract your own DNA using exactly the same experiment. All right, so we're now across the country. We're all in the same space. We've mashed up our bananas, our strawberries, our blueberries. We're going to add the last one, the next step. We're going to add water. So everybody has got water in front of you, but we need one more thing. So the first thing to do is to take the salt that you got and take a pinch of salt and put it in the water. All right, pinch of salt in the water and then gently swirl that water. In the water. All right. All right. Now, what you got is perfect. All right. Swirl that around. Yeah, or you can use this different time. Swirl the water in the salt. Good. There we go. Okay, so this salt is incredibly important. The reason, let me have you attention. The reason this experiment works is that the DNA molecule that we're extracting has a charge. It's like a magnet, right? But that DNA molecule is very negatively charged. But like the end of a magnet, the two ends don't want to come together. And so we add the salt to this water so that we can get all these thousands of strands of DNA to come up and touch each other. All right, so once you've got your salt and water, swirl it around, then who knows what the next step is? Okay, what's the next step? Gently pour the water into the fruit, into your Ziploc. All right, now put the water in and then close that Ziploc. Uh-oh, we need another water.
Okay, so while we're pouring water in there, I'm just going to highlight, we got a couple messages here. We have the grade 5, 6s, and 7s at Tecumseh in Vancouver wanted to highlight that they are using raspberries and kiwi today. If you are using something other than, say, bananas or strawberries or something else we've mentioned so far, let us know what you are extracting DNA from today. And then we'll have to compare at the end and see how these DNA sources are a little different. I'm just going to check back in with Science North there. How are things going there?
Close that Ziploc so we don't spill too much, and then mix it up. This is perfect. Hold it up. Just like this. You want to mix it up so you've got the water and the fruit and a little bit of salt. Nice and well mixed. Okay? If when you hold it up, you can still see chunks of your berries, gently give it a little bit more mass and break that up. Okay. So make sure the Ziploc is sealed, and then set the Ziploc down. And now we're going to make our magic filters. So set your Ziploc down. Okay, zip those Ziplocs. Now I need you to grab your rubber band. Okay, hang on a second. Your plastic cup. And then, who knows what this is? What is it? It is a coffee filter. This is the reason that science gets done on this planet. Coffee. That's it. Full stop.
Dr. Merritt and Lila.
This coffee filter as our DNA filter. So you're going to open up the coffee filter nice and gently, and you're going to put the coffee filter, my darling assistant, you're going to put the coffee filter into the cup and hold it in place with a rubber band.
Dr. Merritt, we are having some trouble seeing some of this on the webinar. I was hoping you might be able to bring some things closer to the camera every now and then. Really, it's just for me. I can't see. That's the problem. Ooh, a plastic cup and a coffee filter and an elastic band.
So we got our cup, our filter, and our rubber band. Lila, will you get our fruit? We're going to take the filter, put it in the cup. Yes, exactly. Mrs. Smith's class, that is perfect. Great job. And we're going to use the rubber band. Let's see. Rich, you're not following along at home. I know that you have the skills to do this. Even you can extract DNA. Actually, Stacy, if we can go to a shot of everybody at some point, that would be awesome. I would love to get a sort of a group shot of all the webinars.
I wish we could. We can't show a gallery view to our webinar viewers, only the classrooms who are joining us live. But
all right, so we've got cup, filter, rubber band, ready to go. All right, Science North, how are we doing on our DNA filters? We're still need a little bit more time?
okay.
All right, so it's really important, hold your filter up. It's really important that that rubber band is all the way around so that when you pour your fruit through it, it doesn't fall off, okay? So take a second, set that filter down and double check. Do you have that rubber band where it needs to be? All right, let's check in across Canada. How are we doing with our DNA filters? Have we got things ready to go?
All right, let's see. We will go first to Mrs. Smith's class, as they had one to show us a couple minutes ago. Mrs. Smith's class, can you show us one of those filters again?
Hey,
You all set? Oh, I like the running.
great. Seven.
Perfect. Okay, let's stay with our British Columbia crew. Let's check out how things are going with Mrs. Allen's class. Excellent. I just need you to say that you're showing me your filters so that the sound causes the video to switch to you.
Say we're showing it.
We're showing it. Yes.
Awesome. Okay, and it looks like we're okay there at Mrs. Wessel's class as well. Does someone have a filter to show us?
If you want to.
They're all awesome. Okay, let us check in with our webinar viewers as well. We have lots of people mashing bananas. At Ms. Holloway's class, they're using bananas and kiwi fruit and strawberries. We also have strawberries in Montreal. We have blackberries. The Squid Squad is using blackberries. And we have a few other classes that are trying strawberries and bananas to see how they might be different. So let's check in again at Science North and see what step we're at here.
So we're going to let that filter through. Whoa, hey.
They look very busy to me. Okay, let's check in with Ms. Burkell's class. How's it going there with your filters, Ms. Burkell's class? It looks good. You look ready to filter. Okay, thanks. All right, let's check in again at Science North. Lila, how's your extract going there? I have a feeling that she can't hear me. It seems kind of loud at Science North. So I'll use this opportunity to invite everyone who is joining us for the webinar and watching, and we can't see you on video. So what I would love to see is if you can share with us some images of your activity today on Twitter. We are following the hashtag FruitDNA as well as hashtag SciLit17 for Science Literacy Week. So please do snap a photo of what's going on in your classroom today and post it for us with one of those hashtags or both. And we might be able to highlight that later in the webinar. We have Brent Peltola from PIR standing by just waiting to see what tweets are going to come through. So please do throughout the activity or at the end show us what DNA you were able to extract. So let's go back to Science North here.
Options. We poured the filter, we filled the, oh, geez, that is, who's got the phone? You poured the fruit through the filter, there you go, and you let it filter the filter. It's filtering through. Now hold it up and look. If you think you've got enough liquid at the bottom, you're good. Now, if, like me, you want to make sure you get that last little bit, you can gently squeeze that filter.
No, they got control.
I think that's pretty good. Right? So at this step, what we need to do is take the rubber band off, and you can either just lift the filter out and throw it away. Let me show you. So this amount of liquid is just about perfect. That's it. That's great. So if you don't want to squeeze, you don't have to squeeze. So just pop the rubber band off and you lift the filter out. If you want to give a little gentle squeeze, you need a back out.
We're part of it.
All right, so we're going to take your filters, take your rubber bands off, and just lift it out. All right, how are we doing? Everybody good? And we're going to lift it out. I'm going to gently squeeze. Oh, you want to squeeze? We're not going to. Oh. Okay,
And just remind us there, Dr. Merritt, what happens if you squeeze too much? Dr. Merritt, what happens there if you squeeze too much? Hopefully we don't find out. I don't think they can hear me. I hear so much activity there at Science North, but I see he's very gently squeezing. We don't want that filter to break.
All right. Okay, so we need you to very organized and toss the fruit out. Yeah. Yeah, so you squeeze from our jurisdiction. Okay, all right, so if you got fruit in there, you leave it and we're going to let it set. Yeah, that over here. Now that's good. So what we're looking for is just like this. All right. Okay, so let's put our DNA, put the fruit down in front of you. If there's a little bit of fruit in there, it's fine. All right, so put your DNA solution down on the table. All right, we're going to let them settle for a second. Now, that DNA is inside the cells. We now have that solution, that liquid in front of you that has the cells, but we need to burst the cells. So the last thing that we're going to add is a detergent. So it's the same detergent that you use in your sink, and you're going to, thank you, you're going to take the dropper, and you're just going to put a couple of drops, just a couple of drops of detergent in with the DNA. Okay, so add just a little bit of detergent. And what we're doing here is.
Okay, let's check in Mrs. Smith's class. Looks like they have something to show us here.
Yeah, we're ready to squeeze out the last of our banana juice.
Excellent. So you're going to squeeze so hard that your filter breaks?
No.
That kind of defeats the purpose, right? Good job.
Thank you.
Yeah, put us on.
Great
All
job.
right. Does anyone else give me a wave from our live classes if you have something to show us at this time, and I'll try to highlight your video. Looks like we have someone at Mrs. Allen's class. Hi there.
We started squeezing the banana into a cup.
So you have some liquid in that cup now?
Yep.
Excellent. So now it looks like you're ready for the detergent. Thanks for sharing. Let's check in Newfoundland. How are things going there, Madame Brunet?
Good. Awesome. Our filter first.
It's a DNA party in Newfoundland.
Yeah, what's not the party?
No, one person at a time so they can hear us. Okay, at this table, what's been happening?
Our filter was compromised.
We've had a filter breakage, a bit of a nuisance. Catching up on it.
You're seeming very not
We're right to wait we got to let's switch to bananas because they're plastic bags. I think it's kind of high energy here. We're not necessarily going to be able to hear ourselves very well.
Okay, well, I will let you get back to your DNA party and we'll check in with you a little later. It seems like everyone is abuzz today. That's excellent, having lots of fun with our activity. Let's check in with Ms. Burkell's class. Maybe they're in Calgary. Where are you guys at with your DNA extraction? Excellent. Okay, let's check back in with Science North. Looks like they might be in between steps. Dr. Merritt, how are things going there at Science North?
What's up?
How are things going there? We're trying to pop in and see what step you're at.
We are swirling. All right, if you've got your detergent mixed into your DNA, hold up that cup. Lots of cups. Perfect. Okay, so we are almost done. You've physically broken up the strawberries. You've filtered out most of the strawberry. If you haven't, that's okay. We'll let it settle down. We've added a little bit of salt so we can coat the DNA molecule with salt. And now we've added detergent to that cup. And that detergent is the last step in bursting open those cells. And so you now have, in that cup in front of you, thousands of meters of DNA. And the last step, we're going to gently pour alcohol across the cup. And where the alcohol and the water meet, the DNA is going to come out of solution to precipitate. So take your alcohol in one hand. All right. Open up the alcohol, hold it up for me so I know everybody's ready. We're getting there. All right, now we're going to take your DNA solution. And we're going to pour the alcohol down the side of the cup. So I'm going to come over here to the camera. So everybody hold your DNA and your alcohol. And we are going to pour it down the side of the cup. Where do you go? All right, now, set that down and look over the top. What you should be seeing in front of you, so pour the alcohol down the side. Let me see. Yes. Yeah, so you pour all the alcohol down the side of the cup. Okay, so now we need our stir sticks. And what you're going to do is gently. Missy. Oh, that looks great.
Yeah, just right up here.
All right, so you want to see that? Just see the two different layers here.
Oh, DNA.
Yeah.
Back up just.
You're going to take your stir stick and you're going to just gently swirl it through.
Perfect.
We're a little bit behind. Can you tell us when you add the salt? Was that
Yes. You can add the salt or the soap either before or after you filter. So you can add them both in your baggie and mush them around, or you can filter it first and then add those and mix before you add the alcohol.
Got it. Thank
And
you.
you're welcome. And just a quick note for everyone, when you're pouring that alcohol very gently into the cup, you want to form a layer on top of your fruit mixture layer. So you want to have 2 layers, and your wispy DNA strands will start to be visible at the interface, or the line between those 2 layers. So I think we might be starting to see some more of that at Science North soon. Or if you have some forming at your location and you're pulling that out with a popsicle stick or a stir stick or something like that, give me a wave from our live classes. And if you're at that stage and you are not one of our live by video classes, if you're joining us through the webinar today, make sure you do share that with us. We would love to see how things are going for you. Just make sure you tweet that photo and include the hashtag FruitDNA and hashtag SciLit17. So let's check in with, let's see who's maybe waving today or who might be ready. I think Science North, we have some more DNA there.
Come closer. Get closer. Okay, back up a little bit. Excellent.
I think that's our longest one so far. Very stringy.
That's a big one. Give it a
Okay, let's check in with Mrs. Allen's class. They're giving me a wave. I think they're ready.
We got DNA, but he knocked it down.
Big one.
Oh my goodness.
It's a booger. It's a booger.
I think you're right. It does look like a booger. You win. Yep. So those were bananas at Mrs. Allen's class, right?
Yes.
We have banana DNA. Excellent. Okay, we're gonna move on to another class. Let's see where else we might have some DNA to show. How are we doing there in Calgary, Mrs. Wessel's class? Yes. How are things going in Calgary with Mrs. Wessel's class? Does anyone have something they can show?
We're just getting started on the methanol right now, so we'll have some snotty substance to show you shortly.
Sounds good. Okay, we'll check in a bit later. Let's see who else we have here. What about the DNA party in Newfoundland with Madame Brunet and Mr. Thorne? Does anyone have any DNA yet? Oh dear. Does anyone have any DNA forming yet? Do we have anyone with 2 layers to show?
Over here. We're pouring alcohol.
Can we bring it up closer to the webcam? Is that possible?
Yeah,
We haven't seen anyone's 2 layers yet today. Let's have a look and see if we can see the layers in the DNA. Just hold it up close and really still if you can.
I'm trying. I can't hold it. I'm short.
All right, I see 2 layers there. Excellent. Oh, I'm just gonna pop back over to Mrs. Allen's class. We've got some more DNA there, and then we're gonna go to Montreal. Stringy bits. More stringy bits in Montreal. Go ahead and say something there at École Lovive.
Right, so you can see the layers separating nicely over here. And in between, that's where the DNA is really visible.
So I can tell you must have been using strawberries there.
Oh yeah.
Excellent.
It smells delicious.
Thank you for sharing. So you can see sometimes you can use a test tube as well. I'm going to check back in with Science North now. They look ready. How are you doing there, Dr. Merritt?
So we're doing awesome. We had some of the high school kids from the high school students extracted from wild blueberries as well as strawberries. So can you show us what you got?
Can you come a little closer?
Okay. Just get closer and hold it up there. So let's see. Not much. Not much because there's a cloud. Yeah. So that's pretty cool. There's a lot less DNA than there is in the domestic strawberries. Okay, that's great. Thanks. And we've got a bunch of strawberries. The camera's all the way up there. There you go.
Oh, yucky.
No. No, right. No. Where's the camera? I mean, stop it. No. Do it, Nikki. It's fun.
Okay, we're going to pop back over. Oh, we have a super close-up of a really, really big glob of DNA at Mrs. Allen's class. Let's see that again.
3 layers. 3 layers.
3 layers. Show me that big glob again.
Oh man, it looks like, I don't know, I don't even.
That's a pretty big glob of DNA. Nice work. Oh, we've got something to show at Mrs. Smith's. We've got Mrs. Smith's class. We've got layers there to show. Awesome. I can see the cloudy stuff in the middle. That's our DNA. Okay, now I know we're still working away in many locations on that activity, but I'm hoping we can transition soon to some questions with Dr. Merritt. Looks like he is running around checking on some DNA extracts. So in the meantime, we will go to Madame Brunet and Mr. Thorne's class. I saw some students trying to show us something there. There you are. You just have to say something for me and we'll see what you have to show.
Hey!
DNA.
That's a pretty big glob of DNA.
Yes.
Send it to everyone, Madame.
And we have
Nice work in Newfoundland. Okay, we are going to check back in with Mrs. Smith in BC. How are things going there?
Great. Take a look at our blackberry DNA.
Oh, wow.
Blackberry DNA.
Make noise so we can be on big screen.
Yeah, there you go. Thank you.
Thanks for sharing. I think you were one of very few groups doing blackberries today. And it looks like everybody's on the move at Science North. Let's see if we can figure out what's going on there.
Okay, that's connected.
Okay. So let's check in. We'll see what everyone is saying on the chat function here. I'm just looking at my chat window. We've got some people who are just at the alcohol stage and some questions coming in. So let's see if we can get Dr. Merritt's attention. Dr. Merritt, can you hear me? I can tell by your run that you could. Are we able to transition to some questions soon? We have questions coming in.
I think that's great.
So we'll just have to make sure that the students at the cavern are a little bit more quiet so we can hear your answers. If we can, if it's possible.
Okay, so we're going to do some question and answers for Cross Canada. Stacey, you want to start us off?
Absolutely. Our first question, let me scroll here. We've got Mr. Yu from Langvin School in Calgary wants to know if meat tenderizer works better than detergent, how critical is the higher concentrated alcohol?
Yeah, so there are a couple of questions there. The meat tenderizer is usually a proteinase, and so it breaks down proteins more than lipids. One of the experiments that we do with saliva, we use proteinase. The other thing you can use instead of meat tenderizer is contact lens solution. It has the same kind of thing. And so those are things you can try at home. So Lila and I have done this by trying different fruits, but you can also try it with different kinds of solutions. So you can swap out the detergent with meat tenderizer with contact lens solution and actually do a little experiment and see which combination breaks things down the best. We had a question here in the cavern about where the strawberries are from. So the reason that we use the strawberries that are in front of you are because these are domestic strawberries that are grown commercially and they're really big. And one of the reasons that they're really big is they have a lot of DNA, and that lot of DNA makes them grow really quickly. And so they're a good fruit for us to extract. We looked at wild blueberries, and those wild blueberries have a lot less DNA. And that's part of being a wild fruit versus a domesticated fruit. So you actually were doing an experiment there to see for about the same amount of fruit, do you get different amounts of DNA? And in the back, what did you find? Was there less DNA in the wild strawberries? Yeah, and that's exactly what we would expect, right? But we actually do these kind of experiments in the lab where we look to see how much DNA are in the different genomes. Stacy, what else do we have for questions?
Oh, we've got multiple questions coming in already. And the next one I think I'll go to is from J.G. Workman in Scarborough, Ontario, asking why extract DNA from fruit, and have you tried extracting from anything else?
Yeah, so we do it with fruits because they're really easy to get, and you can get a lot of fruit. And because DNA is DNA is DNA from any organism on the planet, we're doing the same extraction with anything. Lila and I have done different fruits. We have, in the classroom with undergrads, I do human spit, and you actually get quite a lot of DNA out of spit. So you can do lots of different things. Essentially, every animal on the planet has DNA. It's just a question of how much DNA. And some of them have more than others, every plant and animal. So we tend to choose plants that are easy to get, they don't have diseases, and we get a lot of DNA out of it. It's a great question.
Excellent. Well, next up, we have a question from our party house in Newfoundland over there. We had a question about what you could use that DNA for. Does someone want to maybe ask that question directly?
Right. Oh, it's Daniel's question. Yeah. Sorry, I'm over here in the hidden area. Yeah. So can you use the DNA to like recreate a strawberry? Like could we like lab grow strawberries and feed them in school?
Yeah, so could we use that DNA to recreate a strawberry? At this point, we can't recreate an entire strawberry. But one of the things that we do in my lab is we take pieces of DNA from different places and we put them into an organism and we can create a piece of that organism. So a lot of the work that we do in my own lab, we genetically engineer fruit flies to have a slightly different gene than they started with, and then we look at the biology of that. So we can't yet make an entire strawberry, but we could, for example, make a blueberry that had a little bit of strawberry DNA in it. And that's something we can do right now.
Okay, and next up, it looks like we have a question in Mrs. Allen's class at West Seashelt Elementary in BC. Go right ahead and ask your question.
What would happen if you drank DNA of a certain animal, like a lizard or something like that?
Good
so what
question.
would happen if you drank DNA? So that's a really cool question. It ends up you eat DNA all the time. So anytime, how many of you have eaten a strawberry? How many of you tasted the strawberries in front of you? No, you did not taste the strawberries in front of you. But if you have ever eaten a strawberry, you have eaten DNA. And any plant, any animal that you eat, when you eat that, you actually eat the DNA. And to some extent, you have to do that because we break down the DNA that's in that plant or that animal, and we use that to make more DNA. One of the really cool things that we didn't know about is that when you do that, sometimes the cells in your digestive system actually take up the DNA. And you can find pieces of plant DNA in human cells. It's really sort of odd, but it happens all the time. So that's a great question. So what would happen if you eat DNA? You do it all the time, nothing happens. You actually use it as a nutritional source.
Excellent. I'm going to go now to a webinar question from Tumpane Public School in North York, Ontario. They would like to know if human DNA has a color.
So does human DNA have a color? That's really cool. So when you did the DNA extraction in front of you, what color was the DNA when you pulled it up? It didn't really have a color. And so the DNA molecule itself really doesn't have a color. It might be a little bit whitish, but it's pretty clear. And DNA does not really have a color. And it doesn't matter what organism it's from. It all has that same sort of whitish, clear color.
Great. And let's see where we're going to go next. We have Tecumseh Elementary in Vancouver. The grade 7s want to know if genetically altered fruit will have more DNA. Is that maybe a trend?
No, not necessarily. So a lot of the fruits and vegetables that we grow, we created that line, whether it's a tomato or corn or something else, by hybridizing different species. And those hybrids tend to have a lot of DNA. When you go in and genetically modify it, you're actually engineering the DNA. You don't necessarily increase the amount of DNA as much as you do with things like hybrids. So to some extent, modern genetic engineering will give us a different pattern of DNA than classical breeding like hybridization. And actually, so the strawberries that we use, I can't remember. Do you remember how they're not diploid, they're not triploid? Bananas are triploid. Strawberries are something weird. I used to work on sturgeons. You think strawberries are tetraploid? You're making that up. Okay. So strawberries might be tetraploid. I used to work on sturgeon, they're really ancient fish, and the species of sturgeon that I worked on had 16 copies of the genome instead of 2. So they're really messed up. And they're crickets that have ridiculous samples, or DNA sizes.
Excellent. And we have our next question now from a grade 5-6 class wanting to know why the DNA clumps together.
That's awesome. Why does the DNA clump together? So we sort of talked about that. The DNA molecule is incredibly long and thin. The backbone of the DNA molecule is a series of molecules called phosphate groups. And those phosphate groups have a negative charge. It's like the negative pole on a battery. And so what we do with the salt is we coat that strand with salt to try to hide that negative charge. But it's not completely hidden. And in water, the DNA is completely in solution. So when you had that DNA in front of you, when you were holding up your extractions formula like this, and you just had that red solution, you couldn't see the DNA because that DNA was in solution. When you poured alcohol over the top, alcohol has a different characteristics as a liquid than water does, and it's not as polar. And because that alcohol is less polar, the DNA comes out of solution. And because that DNA does have some charge to it, it clumps together. So it comes out of solution because of the change in liquid characteristics between alcohol and water. You're muted.
I was indeed muted. I'd like to go to a few of our live classes now and check out if they have any questions. We already had a couple. Why don't we go to Mrs. Smith's class in BC? Does anyone have a question there?
Hi. So I heard once that DNA only swirls to the left and not to the right. Why is that?
So that's a really great question. DNA swirls to the left, not to the right. So DNA, like this picture, is a helix. And so honestly, we're guessing that it's a helix, but we're pretty certain that it's a helix. And it is a right-handed helix. And that means that if you held your right hand up and did this, closed your fingers with your thumb in the air, that's the way that the DNA molecule twists. And every DNA molecule on the planet has that same right-handed twist. That's a really neat question.
Excellent. And I'm going to pause here before we go to, we'll go to Mrs. Wessel's class next in Calgary but for a question. Before we go there, let's answer a question from the grade 5s at St. Patrick's School in Ottawa. They want to know what happens if you mix the DNA of a strawberry with the DNA of another fruit.
You get a blue strawberry. It's not true, I'm making that up. So it depends on what DNA you make. So if you just mix it in a cup like this, nothing happens. But if you take pieces of strawberry DNA and try to put it into a blueberry, you can actually create fruit with different characteristics. We're not really good at doing this yet. And so in theory, it's something that we can do. But the reality is what makes a fruit is so complicated that trying to find the right pieces to mix is incredibly difficult.
Excellent. Okay, we're going to go to Mrs. Wessel's class now. Who has a question there in Calgary?
Hey, hello.
Bang, bang.
Do we have a question?
A lot of fun doing this. Can you hear us?
Sorry, do you have a question?
Do we have any questions? Guys, do you have any questions? Guys, come on. Time delayed. Do you have any questions? No.
Okay, we'll come back. You can let us know. Give us a wave if you have a question later. We're going to go to a waving arm in Newfoundland.
We've got a question here, I think, when we come back.
Okay, we've got a question in Newfoundland, and then we'll go to Science North.
Okay. I was wondering if we could without breaking it.
Sorry, you might need to come a little closer. I couldn't quite hear your question.
We were wondering if we could like touch it without breaking it.
Can you repeat the question?
Are we allowed to like touch it without breaking it? Like, would it break apart if we just like touch it?
Can you touch the DNA? Yeah. So don't tell anybody I said it was okay, but yeah, go ahead and touch the DNA. It's actually sort of cool. I think one of the things that you should do with this experiment is actually physically pull that DNA. So that's a neat question. Can you touch it? So we actually have designed this experiment so that the stuff that you use, all the components in this experiment, are things that you have in the house and are not toxic, or not very toxic. You should definitely not drink it, and you should wash your hands afterwards. But one of the things I really would love for you to do is take that DNA at the end of your stick and just feel it with your fingers. You can actually then say you have held genetic material in your fingers. You've held DNA in your fingers. Yeah, once you slow it up, it starts to break up. So you just get smaller and smaller pieces. So it's there, small pieces.
Okay, so what is our question there that you had at Science North in the cavern?
Come over here. What if you put ice in DNA? So that's sort of cool. So what if you put ice in DNA? Then we've got one more question here. So the precipitation, when the DNA comes out of solution, that actually matters how warm the solution is. And so the colder the solution, like the more ice is there, the more DNA comes out, but the more other things come out too. And so you get more DNA, but you also get what we call a really dirty extraction. There are a lot of other things that come out, a lot of proteins and RNA. Right. And we have one more question.
Me too.
Why do some fruits have more DNA than other fruits? So one of the answers to that is we don't know. And so sometimes we do know, like the strawberries that we have here, they have a lot of DNA because of the way that we bred them. But a lot of times we just don't understand why we have different amounts of DNA. And that's maybe a question that you could ask when you become a geneticist.
Okay, so I promised we would go back to Calgary. Let's go to Ms. Burkell's class. They're waving at me here. Do you have a question, Ms. Burkell's class?
Yeah. So potentially, if you use human stem cells and DNA, could you regrow lost organs?
Oh, sorry there, Dr. Merritt. I had you muted. Can you start that answer over?
So can you repeat the question? Sorry, I was.
Okay, just a moment. Go ahead and repeat that question again from Calgary.
Using human stem cells and DNA, could you potentially regrow a lost organ?
Stacy, maybe you can repeat the question.
I can. So using stem cells and some DNA, could you grow a lost organ?
Not yet. But that may be something that we can do in the future. And that's certainly something we would like to do. A lot of the really cool questions in genetics we haven't been able to answer yet. And so, I mean, I'd love to hear those questions, and I'm hoping that the student who's asking them, maybe that's the person that can answer those questions in the future. There's a lot of amazing work that yet to be done in genetics. Questions like that, how do we grow organs, how do we fix genes? We don't know the answer to that yet. But I think we can get the answers to that with people like that asking the questions.
Excellent. Well, I think I'm just going to do a quick scroll here. I'd love to fit in another question or two. Let's go with a nice simple one here. We've got a question from Prince Rupert at Roosevelt Elementary School. They would like to know why the DNA extraction looks like slime.
Yeah. Why does it look like slime? Probably because we don't just pull out DNA, and so there are other things too that are holding on to a lot of the water molecules. But the other thing is that when we pull the DNA out, there's a lot of water that sticks to the DNA, and that water sticking to that long strand gives us a slimy looking extraction. It's sort of cool. It's not slimy.
Okay. One more question from Science North. Okay.
Oh, no, it's the last one. Last question because we have to go.
Oh, we've got to get students to that. This is going to make an answer. But I will make sure I answer your question. Just give me 1 second.
It's a question.
So the question is, can I microwave DNA? I can microwave a cat, but I shouldn't. So you can microwave anything you want. If you microwave the DNA, it'll just break down. So you could, but it'll end up fragmented into lots and lots.
Wait. So just a quick disclaimer there. No one is recommending that you microwave a cat. We'll just put that right out there.
That's sort of the point. You don't need to microwave the DNA, and you don't need to microwave a cat.
Okay, well, we are just going to mute Science North there. They can go ahead and answer questions. We have to wrap this up as we promised we would keep this under an hour, and I've got 1 minute to go. So Thank you so much, Dr. Merritt, for leading our DNA extraction today and answering questions about genetics. All right. And also thank you to all the teachers and the students who joined us today from coast to coast, extracting fruit DNA to celebrate Science Literacy Week. Join us again for more PIR live events, including lab tours, demonstrations, and topics from environmental sustainability to coding to Mathemagic this fall. More information is available at pirweb.org. Thanks again for joining us, everyone, and have a wonderful day.